Welcome to another video tutorial on fluorescence microscope. Okay, so next is the fluorescent microscope. So as you can see in the picture over here, so this is a fluorescent microscope, and so it is connected to the screen, and in the screen or the image produced by fluorescent microscope will have the dark background, and in the dark background you can visualize the specimen very clearly. So it is also used to study the internal structures, and mostly we use this fluorescent microscope in the clinical and diagnostic settings. Okay, so and mostly we used them to detect the microorganisms. Okay, and we use them for detecting the antibodies like autoimmune antibodies which causes autoimmune diseases. And apart from that, yes, we can also detect many other substances rapidly. Okay, so yes, and this fluorescence microscope, no, they mainly work on the principle of fluorescence. So first we should know what is fluorescence. Okay, now any substance, it is said to be fluorescent substance, or it is fluorescent when it absorbs the energy of shorter wavelength radiation and emits longer wavelength radiation. That means there is a substance, and if that substance is able to absorb the shorter wavelength radiation, and if it is able to produce the longer wavelength radiation, that substance is called as fluorescent substance. Okay, now let me explain this with the help of a diagram over here. So yes, now this is the atom of the fluorescent material or fluorescent substance. Okay, what happens here is, see, this is atom, there is nucleus in between, there are orbits around, there is electron over here. Okay, this yellow color thing is electron. Now what I'll do is I will produce the shorter wavelength radiation here. Okay, so see, the color of the radiation over here is blue color. So blue color will usually have the wavelength of around 450 to 495 nanometers. It is shorter wavelength. Okay. So when I produce this wavelength radiation over here, it absorbs the radiation. Okay. And so the electron which is present in the lower energy state over here will get excited to the higher energy state. It will jump to the higher energy state. See, electron is kicked up to the higher energy state. Okay, and the electron over here cannot remain in the higher energy state for a very long period of time because it is very unstable, and as a result it will jump down to the lower energy state. And when it is jumping down to the lower energy state, it will produce some energy or light which is of shorter wavelength, I mean sorry, longer wavelength. Okay, so as you can see, this is a green light, so green light will have longer wavelength. Okay, it got excited because of shorter wavelength light, and now it is producing longer wavelength light. And this green light knows it will have the wavelength of around 495 to 570 nanometers. So now as you can see, this is a longer wavelength. The wavelength over here, the wavelength of green light is longer when compared to the wavelength of blue light. Blue light had a shorter wavelength, right? So this process over here is called as fluorescence microscope, or it is called as fluorescence. Okay, and the thing is, in our nature there are many materials which are naturally fluorescent. Okay, for example, substances which have chlorophyll, no, so they're naturally fluorescent, and they'll be able to produce natural fluorescence, whereas there are many other substances in nature which are not fluorescent. So when you want to absorb the specimen which is not fluorescent, what you need to do is you need to tag them with a fluorescent dye. We get a dye which is called as fluorescent dye. You need to tag them with a specimen, and then you need to observe under this microscope. Okay, so just like in bright field microscope, we stain the specimen and then we visualize them under microscope, right? So just like that, in the fluorescent microscope, you need to tag them with a fluorescent dye and then you need to visualize them. So what happens when you tag them with a fluorescent dye? So they usually undergo fluorescence. So they will be able to produce and absorb the light of a particular wavelength, and as a result you can observe the specimen very clearly. Hence we use this fluorescent dye. So the most commonly used fluorescent dyes are DAPI, acridine orange, oromine rhodamine, Alexa fluoresce, and dye-light 488. So these are some of the commonly used fluorescent dyes. Okay. Then okay, so next is the components of fluorescence microscope. So as you can see in the picture over here, so these are the components of fluorescence microscope. Okay, the first thing what you need to know here is the specimen which we are using here is tagged with a fluorescent dye. Okay, as I discussed in the previous slide, so fluorescent, once you tag them with a fluorescent dye, what happens is they will absorb the wavelength which is shorter and they will produce the wavelength which is longer, and that is called as fluorescence, and only the fluorescent light will be sent to the detector and you can visualize them under your eye or in the screen. Okay, so hence you should know that the specimen will be tagged with a fluorescent dye, only then it will be able to undergo the fluorescence over here. Okay, so the first component over here is the light source. So the light source here is the LED, that is light emitting diode. So and it emits at specific excitation wavelength. Okay, and so after the light source over here, so next is the excitation filter. So this is the excitation filter, as you can see here. So this is the excitation filter, and so this excitation filter, what they do, so they allow only the excitation wavelengths to pass through. Okay, ensuring that the fluorophores are excited efficiently. Now what happens now, as you can see here, so from the light source all colored wavelengths are getting produced, but this excitation filter, so they allow only one wavelength to move out, only one wavelength to pass through the specimen. Okay, as you can see here, only blue light is coming out from the excitation filter and only the blue light is moving to the specimen. So here all the colors are there. From all the colors it is only sending the blue light to pass through the specimen. Okay, so hence, so this excitation filter allows only one wavelength to get excited. Okay, and then it passes to the specimen. After that, next is the dichroic mirror. Okay, this is a dichroic mirror. What does it do? Why do we call it as dichroic? Dichroic means it, see, what will it do? It is, it is positioned near the objective lens. The dichroic mirror reflects the excitation light towards the sample while allowing the emitted fluorescence to pass through. Okay, now yes, excited light, that means it has both the roles over here. Dichroic means it allows the excited light to pass through the specimen and it allows the fluorescence light to pass through the detector. So specifically it takes one wavelength of light and it is passing, and it is sending another wavelength to the detector, hence we call it as dichroic mirror. Okay, now what happens is, yes, this blue light now it is moving to the dichroic mirror, and from the dichroic mirror it is moving to the objective. Okay, so and what does this objective do here? So yes, objective collects the excitation and emitted light and it determines the resolution and it creates it, it gives the magnification for the microscope. Okay, now the blue light is getting passed through the specimen, and so now it is getting fluoresced, and now the fluorescence light is green light. As you can see here, the fluorescence light, green light, is getting passed through the objective, and from the objective it is moving back to the dichroic mirror, and from the dichroic mirror it is moving to the emission filter. Okay, so now what does this emission filter do? So this will selectively allow only the emitted fluorescence to pass through the detector. That means it only allows the fluorescence material, that is fluorescence light, to pass through the detector. It will not allow the blue light over here. It will allow only the green light, that is only the fluoresced green light only it will allow to the detector. It will block the excited light, that is blue light will be blocked over here. Okay, and lastly, as you know, the detector, so detector is the one which captures the emitted fluorescence and then it converts to the electrical signal which you can see through the screen. Okay, so this is the components and this is how it works. Okay, and simple if I want to tell, yes, So this is a light source which produces the light of different wavelength, so which will go to the excitation filter. This excitation filter will excite only one colored wavelength, that is, it only sends the blue light over here. Blue light will move to the dichroic mirror, and then it moves to the objective, and then it moves to the specimen. And in the specimen, yes, fluorescence is happening and green light is produced, and this green light is moving to the objective and it is going back to the dichroic mirror. Dichroic we tell because it specifically sends one wavelength of light, and it takes back another wavelength of light to the detector, so hence we call it as dichroic mirror. And from the dichroic mirror it goes to emission filter. This emission filter only allows the fluorescence light to pass through the detector, while it blocks the emitted light, that is, it blocks the blue light and it only allows the green light to pass through, and then it moves to the ocular, and then finally it will be sent to the detector. Okay, so these are the components. And then okay, next we will study on the applications of fluorescence microscope. And as you can see here, so these are two pictures which is produced by fluorescence microscope, and you can see how beautiful it looks. Okay, so the background is completely dark, and so very clearly you can visualize the internal structures. And it is that not only the green light will be produced for fluorescence. Okay, so you can just select the wavelength of light, and accordingly the fluorescence may happen. Okay, and you can also get red color, blue color, depending on the wavelength. Okay, and yes, the cell biology, studying cellular structures, organelles, and for interactions within the living cells, we use this fluorescence microscope. And apart from that, in immunology, so to study specific molecules, antigens, like antigens, antibodies, we use this fluorescence microscope. And apart from that, gene expression, protein, for protein localization and interactions, we can use this microscope. And in neuroscience, for neurological pathways, for neurotransmitter activity, and for calcium signaling and all those things we use. And then in microbiology, is mainly for studying the microorganisms, we use this microscopes. We can also study their behavior here. And for medicine, like diagnostic imaging, and including diseases like fluorescent labeling, all these things are the applications of fluorescence microscope. And then okay, so next is the limitations of fluorescence microscope. Yes, so fluorophores gradually lose their ability to fluoresce as they are illuminated in photo bleaching. Yes, so photo bleaching means when this fluorescent dyes, no, when they're exposed for a longer period of time under the sunlight or under the light, so they lose their property of fluorescence. Okay, that's one limitation. And then apart from that, they also generate molecules such as reactive chemical species. And as we all know that reactive chemical species, they are harmful for the cell or the specimen, right? So hence this is another limitation. And apart from that, so fluorescence microscope, no, they allow only observations of specific structures labeled for fluorescence. Now for example, you use the fluorescent DNA stain. Okay, you use the fluorescent DNA stain and you tag the specimen. So then what happens, only DNA you can see, it reveals only the DNA, whereas you cannot find other cell morphologies. Okay, so all these are some of the limitations of fluorescence microscope. Okay, then